Welcome to The Explainer. Today, we're cracking open a fascinating clinical briefing to uncover the real science behind biological age testing. Look, if you've been paying any attention to health optimization lately, you've definitely heard claims about tests that can tell you how old your cells truly are. But here's the secret. Not all of these tests are created equal, and honestly, the underlying molecular biology is often completely misunderstood. We're going to clear all of that up today by going straight to the diagnostic data. Here's our roadmap for today. We'll be hitting the biological age problem, the epigenetic aging paradox, isolating the true signal, models and algorithmic weighting, tracking clinical health trajectories, and we'll wrap it all up at the final executive synthesis.
All right, Section 1, the biological age problem, why global sums fail.
Let's dive right into the foundational problem with how most of us view genetic aging. The core thesis here is that targeted CpG panels succeed where global methylation absolutely fails. See, for a long time, the massive misconception was that you could just tally up the total amount of methylation across your entire genome to figure out your age. But as this briefing reveals, that approach is fundamentally flawed.
It all comes down to what researchers call the initiation question. If DNA methylation generally goes up as we age, why is global DNA methylation such a bad tool? Well, total DNA plus total methylation does not equal biological age. Taking a massive zoomed-out sum of everything happening in your genome literally destroys the clinical value of the data.
And here is exactly why that matters. Global methylation carries a staggering margin of error of plus or minus 10 years. Just think about that for a second. If your doctor runs a test and says, well, your biological age is somewhere between 30 and 50, what do you even do with that? It's virtually meaningless for tracking your personal health trajectory.
Which brings us to Section 2, the epigenetic aging paradox, signal versus noise. To understand why that error margin is so incredibly huge, we've got to zoom way in to the cellular level. DNA methylation is basically just the addition of a methyl group to your DNA. Think of it like a tiny biological padlock. It doesn't rewrite your actual DNA sequence, but it locks it up to alter or completely silence gene expression. It's essentially telling your body to either read a certain instruction manual or just ignore it.
But here's the kicker. Location changes absolutely everything. If that methyl lock lands on a promoter region, transcription factors get blocked and the gene is aggressively shut off. But if that exact same lock attaches to a coding region instead, the polymerase just glides right past it and normal transcription still happens. The physiological impact depends entirely on exactly where that lock is placed.
And this perfectly illustrates the epigenetic aging paradox. As we get older, targeted gene silencing, those crucial locks on specific CpG islands, actually goes up. But at the exact same time, overall genomic instability causes a systemic loss of global methylation, so it's crashing down. You've got two totally opposing biological phenomena happening simultaneously. If you just sum them together, they completely cancel each other out and boom, you've destroyed your diagnostic signal.
Plus, a global methylation score picks up a ridiculous amount of genomic noise. Just a standard blood draw has shifting ratios of different white blood cells that radically alter your baseline. Got a temporary spike in inflammation or fighting off a passing bug, that spikes the volatility. Even evolutionary garbage DNA like line 1 elements, which make up roughly 17% of our genome, will totally skew the results. All this daily chaos easily drowns out the real aging signal.
Moving right along to Section 3, isolating the true signal, finding the clocks. So how do clinicians actually get an accurate reading? By isolating the signal from the noise. Scientists target what are known as CpG islands. These are hyper-concentrated clusters of cytosine and guanine that act as supersensitive biological clocks. Unlike the rest of the genome that fluctuates wildly over a common cold or a stressful week, these specific promoter regions steadily accumulate methylation at a highly predictable, age-correlated rate. They're practically immune to that daily environmental static.
This leaves us with three main tiers of detection today. First, global testing. It's super cheap, but as we just learned, it's got that useless 10-year error margin. On the other extreme, next-generation sequencing maps the whole genome. It's incredible, but it's extremely expensive and mostly reserved for hardcore research. The sweet spot in the middle? Targeted GPC panels. This is the true clinical standard because it captures those precise aging signals at a moderate cost, completely bypassing all that whole genome noise.
Okay, Section 4, models and algorithmic weighting, making sense of data.
Once we actually isolate the data, we have to compute it. You can't just count up the methyl groups and call it a day.
Epigenetic clocks rely on highly complex mathematical algorithms. Why? Because the genetic sites are not created equal. Certain locations carry vastly heavier diagnostic significance than others. Because of that, they need specific algorithmic weighting to actually calculate a valid biological age. To put that spectrum into perspective, let's look at a forensic model versus a clinical model. If law enforcement finds a sample and just needs a rapid, rough age estimate of a suspect, they can use a minimalist 3-site algorithm. It's fast, but definitely limited. But if you're looking for a deep clinical health check, doctors rely on something like the famous Horvath model, which averages and weights a massive, comprehensive array of 353 distinct genetic sites.
Let's get into Section 5, Tracking Clinical Health Trajectories, the Real World.
Now that we understand the molecular biology and the math, how are medical pros actually applying these advanced algorithms for real patients? It brings up a vital question. If even the absolute best tests can have some background static and a single number isn't the whole story, how does your doctor use this data to actually improve your health and longevity?
The answer lies in the huge difference between static numbers and dynamic health. In forensics, the goal is just chronological age, a single static snapshot. Guessing a suspect is 48 plus or minus 3 years. But clinically, doctors don't care about static snapshots. They want to identify biological age discrepancies to assess your true underlying cellular health and metabolic risk. They're looking for your dynamic health trajectory.
Essentially, doctors are tracking the delta. Think of it like a pediatrician tracking a kid's height curve on a chart. One single epigenetic snapshot is really just a dot on a graph. The true medical value comes from tracking the rate of your aging over time, especially after you make lifestyle interventions. Let's say a patient starts out with an accelerated aging slope, but a couple of years in, they implement a strict diet change and completely stop drinking alcohol. Suddenly, their rate of biological aging drastically slows down. That flattened curve, that change in the slope, that is exactly what your doctor is looking for.
Finally, Section 6, the final executive synthesis, putting it together. Let's tie all this complex molecular biology and clinical strategy into a clear, unified conclusion. First, why do these clocks correlate so well with overall vitality? It's due to a devastating 4-step genomic cascade. When you lose methylation locks, garbage DNA gets expressed. Your immune system then attacks those weird proteins, which causes systemic inflammation, and that leads straight to cellular exhaustion. Oh, and a quick note on the telomere disconnect here. Some people think measuring telomere length is enough, but it is completely insufficient. Did you know active cancer cells can extend telomeres perfectly, or that patients in a vegetative state often have excellent telomeres despite having zero clinical vitality? Telomeres simply do not give you the full clinical picture that targeted epigenetics do.
To summarize this briefing brilliantly, just remember these 3 pillars. Number 1, location over quantity. Global sums are out. Clinical accuracy requires isolating specific promoter regions. Number 2, algorithms over raw data. Not all loci are equal, so distinct mathematical weights absolutely must be applied. And number 3, trends over snapshots. A single number will always be prone to noise, so the real clinical utility comes from tracking the rate of change within the exact same patient over time.
We know now that your epigenetic clock isn't a permanent ticking countdown. It's a dynamic system that responds directly to how you live. So I'm going to leave you with this. If you could precisely track the rate of your cellular aging, what lifestyle interventions would you choose right now to rewrite your biological trajectory? Thank you so much for joining me for this explainer. Keep optimizing, and I'll catch you next time.